Good evening, everyone. Welcome to BioVanguard CellTalk presentation, round one. For today's presentation, we have Madhuri Bhattacharya as our presenter, and she will be presenting on the topic telomere length and cognitive changes in 7,877 older UK adults of European ancestry. Before starting the actual presentation, let me introduce Madhuri Bhattacharya. Madhuri holds a bachelor's degree in microbiology from Ramlal Anand College, University of Delhi, and is in the final semester of her master's program in nutrition biology at the Central University of Haryana. Her academic journey has honed her expertise in microbiological techniques, food quality analysis, and the core principles of nutrition science. Through this fellowship, she is looking forward to acquire advanced scientific and research qualities, which will help expanding her knowledge base and empowering her to contribute to high-impact scholarly publications as well as aiding in thesis writing. Now, I warmly welcome Madhuri to give her presentation.
Okay, good evening everyone. My name is Madhuri and here is my presentation. It's on the topic telomere length and cognitive changes in 7,877 older UK adults of European ancestry. So before presenting, I will give a little background study of what is telomere and before that, what is aging. So aging, as we know, that it is a mixture and amalgamation, if the word is correct, of different psychological metabolic changes we can observe in different stages of life, and ongoing that aging occurs. And telomere shortening is a very important part of, as you can see, in aging. And along with aging has a consequence, which is cognitive decline, which we can see in dementia, in the form of Alzheimer's. Parkinson disease is a genetic basis, but little bit of memory changes, reduction of many of the major events of life, they tend to forget. So in this study, what the scientists want to observe is that if there is any relationship between telomere shortening and the cognitive decline, as we can see in the patients having who are older in age. So telomere, here the introduction is given. Telomere are DNA and protein complexes that cap the end of chromosome that minimizes the each cell division. With each cell division, with age, telomere gets shortened. And here we can see in the image as well that there is cap in the ends of the telomere, in the chromosome where the telomere is present. Few studies have investigated the relationship between genetically predicted telomere, cognitive decline, and age-related dementia, but mind you, those results are not very correct. Like some say that yes, there is some relationship, and some say that no, they are independent of each other. Now, telomere length can hence be treated as a reliable biomarker that can help in early detection and monitoring of cognitive health. Previous studies had a less number of participants, therefore creating need for more large-scale longitudinal studies to explore this relationship. Focus of current studies to identify if there is any SAID association by taking both telomere length and measure telomere length and genetic disposition for telomere length and predict dimensions of cognitive performance in a longitudinal sample of older UK adults. Genetically predicted, here the number is given, 7,877, and measured one were 846 telomeric length was assessed for its association with initial cognitive behavior and changes in cognitive performance over time, which are given with the different tests here given, and I will explain it later during the presentation using latent growth models. Hypothesis of this study was longer telomere length measured and captured by PGS-TL would be related to better performance on the cognitive test at baseline and over time. So these are the different references, what they have during the literature review, what they have understood, that several potentially modifiable factors have been identified that may influence telomere length, including physical activity and educational background. Mendelian randomization studies have suggested a possible causal relationship between telomere length and cognitive function and decline. Inconsistent results are present as compared to previous studies. So here is the methodology, how they have begun the study. So there is an inclusion criteria of enrolling. The Protect was the this is the organization which studies on the older adults for any cognitive related studies. They have included more than 50 years old patients, no diagnosis of dementia, and there should be an access to computer and internet. And they have so here are the studies. Here are is as the image is showing that they have taken some people, and the number is given 7877. Polygenic scores for telomere length, which is the test for PGSTL, calculated using genetic data. Relative telomere length was quantified using DNA samples and a modified version of quantitative polymerase chain reaction protocol. So here the image is given, and they have taken DNA from saliva sample, and genotyping is done. Then low genetic score, high genetic score. PGSTL was one test and RTL was a different test. And through them, what they have done is they have done baseline study, year 1, year 2, year 3. They were the different years in the through little bit of time gap. They have done this test. Now cognitive performance, how it was assessed, they have taken 4 different online cognitive tests, which was first was paired associate learning, tested visual spatial memory and learning, digit span test for working memory, self-ordered search, executive function and spatial memory, and verbal reasoning test. And the battery test, I was as I was explaining, 3 times over a period of 7 days they have done. Now, for statistical analysis, they have done using latent growth models through 2 tests, conditional model and unconditional model. For unconditional model, it has been given helps to examine the natural growth pattern by not considering the external variables. And conditional model, they have predictor and covariates, so they are comparing on the basis of that. The sample was further divided into 2 groups based on the median age to compare results for middle-aged and older adults. So here are the results. Now I will be covering the results. So the prediction of cognitive trajectories, unconditional model, fitting done for unconditional model, that is without the intercept and slope factors, condition on predictor or covariates, for each of the 4 cognitive outcomes to determine the pattern of growth. Unconditional model fit statistics suggested that linear models provided an acceptable fit. So here was the acceptable fit to the data for the paired associate learning and digit span test and self-ordered search task. Whereas for word task, a quadratic model was a provided acceptable fit. And next, what they have done, I will be explaining more on that. Prediction of cognitive trajectories, whole sample with PGSTL. Fitting then done for conditional models to explore whether PGSTL or RTL predicted baseline cognitive level, that is latent intercept, or change over time, that is latent slope factors. PGSTL did not predict baseline cognitive performance or change over time or any on any of the cognitive tests after adjusting for covariates. So here is, as per the graph, a prediction of cognitive trajectories subsample with RTL data. RTL also did not predict baseline cognitive performance or change over time on any of the cognitive tests after adjusting for covariates. So the genetically predicted telomere length predicts change in verbal reasoning. Though effect size were small, individuals with higher PGSTL initially experience slower improvements on the verbal reasoning task. So, as per the graph here given, that high PGSTL, which is shown by the blue line as per the graph, it shows high level and experience slow improvement on the verbal reasoning task only, followed by a more rapid increase in performance over time. And the next graph, which shows longer RTL predicted worse performance on the verbal reasoning task at baseline level, but did not significantly predict change over time. Results remain consistent when analyzed in subgroups. That is, means there is no further change or any other data was updated. Example, older versus middle-aged adults. In case of middle-aged adults, which is less than 62 years old, no significant association was observed. So conclusion here, as I have understood by this data, was that there was no relationship between telomere length and cognitive decline. First study to examine, but yes, this is the first study to examine the relationship between telomere length and longitudinal cognitive performance in normative aging using both measured and genetically predicted telomere length. Finding here was neither measure or telomere of telomere length, as I have said before, that there was no relationship. Overall, the results drew into question the utility of telomere length as an aging biomarker that is capable of predicting cognitive performance. Future direction, as I have said, the study had some limitation, which we can observe that the current sample showed evidence of participation bias, such as being more highly educated, because these participation, they have mentioned that they were more highly educated than just general population, so that can affect the telomere length and the study as well. And some other tissue sample can be taken, like here they have taken just saliva sample, so they could have taken some better sample as well. And as Ananya mentioned, epigenetic clocks can be a reliable methodology to be used rather than telomere length. Other variants like genetic and environmental factors can be taken into consideration. So yes, that's it. This is my reference, and thank you.
Thank you, Madhuri, for your presentation. Audience, you can type any queries if you have any queries in the chat box. Yeah, we have one question from Vijay sir. Did they look at baseline telomere length of participants?
Yes, they have taken.
Okay. Yeah, I think there were no more questions. So thank you, Madhuri, for your nice presentation. Thank you so much. Thank you everyone for attending the CellTalk presentation.